I think we've got a good group here, so we can go ahead and get started. Um, so just so everybody knows, Mackenzie's going to be recording our session today. Um, and I'm going to share in the chat our running agenda and meeting notes. So if you get a chance, please sign in there so that we know who was attending the meeting today. So I'm going to share my screen. Okay. And get us started. All right. Everybody see okay?
Yes,
Fantastic.
thank you, Julie.
Thank you. Okay, so thank you for joining us. As you know, today we're going to be continuing our discussion of benchmarking using our updated predicted effector gene matrix metadata standard proposal. Um, so this is just a brief summary of our agenda today. We're going to start by going through just a brief introduction and some logistics for promotion and planning of our ancillary session at ASHG. Then we'll go through a summary of the benchmarking activity. We're lucky to have several volunteers from this working group who participated in this activity. So we're really excited to showcase your results and hear what you as members of our community thought of the standard and how we can make it better. Um, we'll then go through how we can fit this PEG matrix standard to a PEG list standard. And Aoife and Laura are going to present that. So we'll start with just a quick introduction. So just to remind you, this is our proposed timeline for this effector gene working group. So we started, with our second meeting back in June after a kickoff meeting in May. We initially sent out a version 3 standard and template of our straw man with some feedback from all of you, and then have built on this iteratively so that we can expand the straw man to accommodate for various needs of you as the community. So our most recent version that we produced was generated in early August. This is version 3.1, and this is what we have used for our benchmarking activities. That brings us to our meeting today. And then finally, we're going to have one more meeting in early October before ASHG, which is October 14th through 18th in Boston. So if you're headed to ASHG, we wanted to promote a couple of things that we have going on there. We're going to have, as always, a exhibition booth for our Knowledge Portal Network. This is booth 1426, and we'll be exhibiting there October 15th through 18th, 9:30 to 4:30 each day. So if you're there, we would love to see you. We would love to have discussions with you, so come visit us there. And then also, we're really excited to announce that we've secured a date for our ancillary session at ASHG. So this is a predicted effector gene workshop, which we're having Friday, October 17th from 11:45 to 1:15 in room 259A of the Convention Center. Um, so this is our preliminary agenda. We're going to recap the sort of landscape of predicted effector genes that we had when we came into all of this. We'll introduce the new standards that we as a working group have collectively defined, give some presentations on the benchmarking activities, and then finally have a lot of time for group discussion and review of the standards. And our hope is that we can get some of the folks who are here, on our working group calls regularly, but also some other members of the community to attend this session and give us their feedback on how to make these standards more broadly applicable. So we have a couple of asks for you. First, if you could fill out this doodle poll by this Friday to let us know when you're available to attend our October test run for this meeting, that would be great. Um, since this is coming up relatively quickly, we wanted to just use this as an opportunity to go through our how we're going to present our standards to the broader community and get your feedback on that. So if somebody could paste this in the chat The link is available in our running agenda, and folks can fill that out. Our second question, I'm going to stop sharing so we can see the answers. Will you be attending ASHG 2025? If you could please raise your hand if you are. We can get just kind of a quick head count. Okay, so Sylvanus, great. UA, I'll be there. Anyone else? Okay, so for those who are attending, it would be great if you could register. So Mackenzie just pasted a link to the form um, in the chat here. So as she said, this event will be free, and our goal is for everybody to be there who can. So um, just fill that out when you get a chance.
Can I add, Julie? We also have a poster the day before
Yeah,
on the Thursday afternoon.
yes. Fantastic. Thank you. Okay. Apologies, everyone. I'm gonna get my slides back up here. Okay. So, I'll share again. Here we go. Okay. All right. And then finally, if you could please share this information with your networks, Mackenzie is going to send out a more official sort of save the date. And we'd appreciate if you could circulate this to anybody that you know so that we can get other folks to attend this as well. All right. So now we're just going to briefly summarize what this benchmarking activity is. We've talked about this at length at our previous meetings, but we just wanted to refresh your memories on what the objectives were here so that we can start getting into the results. So as you know, initially we manually had curated 169 peg matrices and reviewed them to identify their common features and proposed a prototype standard that we thought would perform well in practice. You all, over the last couple months, have helped us to iterate on this initial prototype to produce our most recent version, which is version 3.1 of this strawman standard, as we're calling it. We then recruited individuals from this community to sort of test drive this prototype standard on actual data and share your experiences and suggestions. And then finally, with your suggestions, we're going to improve this prototype and share it with the wider community at ASHG and beyond. So this is just a reminder of how we defined the PEG matrix metadata standard for our current version. We started by extracting key entities. We then defined the relationship between these entities to define the attributes of the key entities and which of them should be mandatory versus optional. So for instance, variant level information should be mandatory, and we defined what the mandatory information for each variant would be within that matrix. And then finally, we defined a schema in YAML format that could be used for this new metadata standard. So for this activity, we asked that a group of volunteer benchmarkers start by selecting a starting peg matrix or matrices if they chose to do more than one. They could either bring their own or choose some that we had previously defined. We then asked that each of our volunteers reformat the matrix using a suggested standard. So this is, again, the version 3.1 standard, which included a template and some standardized columns. We asked that each volunteer choose one sort of sample trait for this benchmarking activity. We didn't want people to feel like they had to go through every single trait in a GWAS, for instance, but thought that this would give an example of how well the standard worked. We asked for each individual to log their sort of steps that they took in reformatting, share scripts where available. and then document any difficulties that they encountered. Um, so finally, we ask that everybody suggest improvements to the standard so that we can refine it moving forward. All right, so again, there were two options here. Benchmarkers could either bring their own data, so these could be variants from GWAS, variant or gene-centric evidence, and then metadata and context that accompany them. And then we provided the updated standard, which we most recently revised after our last meeting in July. A template to help get started, suggested mandatory column names and suggested column names, and then clear instructions and definitions for each evidence type. Um, as we mentioned, we had some sort of sample Peglet matrices that individuals could use if they chose, and those are provided in our knowledge portal network. So we're fortunate to have a team of volunteers with us from multiple sites. So Matt Paul, who couldn't be with us here today, but did a wonderful job on some benchmarking, and we'll just briefly share some of his results. Abdurrahman Shianbola and Aise Demirkan from University of Surrey. Simon Siskowski and the Open Targets team from the Wellcome Sanger Institute and UAG from EMBL EBI. So um, our hope was that collectively each of these benchmarkers would choose a different type of PEG matrix and have a different sort of set of experiences coming into this that they could then share with the team. All right. So I'll start with a presentation of some of these benchmarking results. These are from Matt Paul from CHOP. Um, so Matt chose to do some benchmarking using this medRxiv article here. So this was a fine mapping study across 148 complex traits derived from three large-scale biobanks from the Biobank Japan, FinGen, and UK Biobank. He selected agent monarchy as a trait on which to do this benchmarking. Noted in his sample YAML file that these were individuals of European ancestry, and then noted information about the nature of the primary variants as credible set lead variant, and then additional information for each level of evidence that we required here. So for the variant, gene, locus, and then types of evidence for each. So notably within this study, he focused largely on types of evidence derived from chromatin interaction data. So he had a lot of Hi-C data and other information of that sort for the variants. So these are just a summary of some of his comments from this activity. He said that overall the format seemed reasonable and required about the same amount of effort as would be required to deposit data to Geo. Um,
निकाल रहे थे हम वही बोले हमारे लाल क्यों दिखा रहा है टीम से चलो
I'm sorry, did somebody have a comment? Okay. Um, all right. So he said that he thought the YAML format was pretty easy to to follow and covered most of the cases. He did note some issues that came up when doing this, so we thought this was great and made note of these. So he said that for some of the columns, they seem like they're transformations that don't cleanly sort of fit the criteria. So for instance, some of the max results for a variant specific score would put them more at the locus level than at the variant level that we were actually asking. He said that there were some areas that were unclear on the current design in terms of where the data should be added in the metadata table. So that's something that we can work on revising. And then finally, he said that a lot of the information necessary for the table are sort of described in other papers. So a frequent use case is for authors to generate a new data set or method for prioritization and then provide the scores from other data sets for supporting their predictions. He thought that we might include a metadata entry for the columns in data sets that are dependent on other columns. Um, we asked, could you assign columns into the strawman V3 sections? So these were variant gene locus, gene-centric evidence, variant-centric evidence, and integration. His answer to this was yes, but he thought that the variant assignment was a little bit fuzzy. And some of the features would normally be considered like variant level features, but the table didn't really take the results from the individual variants. Um, and finally, we asked, could you assign all the evidence streams to the strawman V3 evidence categories? He said it was challenging to track down the evidence that the authors had used from the prior peg list, um, and didn't think that it should be so time consuming if authors are uploading their own data. He also thought it was unclear on how to format some of the additional columns for gene annotations. And so we might treat these as gene-centric evidence or have like a more standardized gene entry. We asked if the source material included gene-centric and variant-centric evidence and whether these were presented together or separately. And he said that for this data set, most of it was gene-centric and some was variant-centric. So a lot of the evidence was from resources that included multiple cellular contexts, like subsets of tissue or blood cells. But most were reported as the max value without actually providing that cellular context. So he thought that including cellular context should actually be required as a part of the evidence here. And finally, we just asked for other comments. So he said for chromatin conformation assays, might be useful to include resolution as an option. So for instance, if you have restriction enzyme elements, you could have, specify whether you're using a 4-cutter or a 6-cutter, and whether, how these were binned computationally in terms of the analysis. Um, he also said that there are other assay-specific pieces of information outside of, just chromatin conformation assays that might be useful to consult with individuals in those fields to see what should actually be required within those types of evidence. So, for instance, if you're doing MPRA, you might want to have the number of barcodes contained within there. So, this is something that would largely be dependent on each individual use case and would require some additional feedback from experts in those communities. Okay. Sorry, I'm just going to advance my slide here. It's stuck. All right. With that, we can go ahead and move on to our next benchmarker. Abdurrahman, I know you had done some benchmarking. Would you be willing to share your results?
Yes, absolutely.
Great, thank you.
Need to share my screen. Can you see
While
my
you're
screen?
getting ready, can I make okay, you're ready. You ready?
So this is the benchmarking activity of the migraine predicted effective gene. That's the assignment we picked. Myself and Dr. Aisha of the University of Surrey. So we got the different information from different sources for the data. And for example, we got the variant information and the gene-centric information from the PEG knowledge portal, and the rest of the columns from the GWAS catalog. I also found that the column names are not consistent across with regards to the published paper or GWAS catalog or the knowledge portal. Same with the YAML, the metadata. One major challenge I found with this is that some of the columns, standard columns in the YAML, is not compatible with the information with regards to the particular trait, the migraine disorder. So the GWAS source was from the GWAS catalog, and that's the IED. We got the trait information from the GWAS catalog as well. The variant information was from the PEG knowledge portal. So one thing generally is with this is the PEG knowledge portal has got the gene-centric evidence information, but to get the variant-centric information, I had to look it up in the GWAS catalog and somewhere from the published paper as well. And I also found that the good thing is the PEG knowledge portal is actually compatible with the information on the published paper. So it looks like the information was sourced from the published paper. These are the list of the problems I encountered. As mentioned earlier on, it's the missing columns in the knowledge portal, and I had to go into, at least to fill up the whole data, I had to look at minimum of 3 different sources, including Ensemble as well. I found that YAML, and apologies, it could be because of my background. I've got background in computer science, so I've got limited biological knowledge. So it could be because of that I found the YAML difficult to customize, as the standard columns do not match the information I sourced in terms of the key value pairs. I assumed we could make the knowledge portal a bit more compatible with the GWAS catalog, with the YAML as well. And getting the trait ID wasn't that straightforward. I think we eventually got that from the GWAS catalog. I just thought to give some recommendation, if possible, given my experience and background, and also the past few months as a PhD candidate looking into genomics data and tools. I would suggest we propose more entities to meet some other assignment, like for example, the migraine predicted effect genes. It does look like different traits have got different entities that are compatible with them. I thought there could be a tool that could simplify the whole process, and we could get a Python script, for example, that would take in input data from the mappings and reference different sources. One such source could be Open Target, GWAS catalog, and so on. And that could automate the generation of a data file and a metadata that would create some consistency as well, and will reduce stress in terms of the complication in creating the time it takes to create that data, and hopefully would encourage the adoption of the recommendations by the working group. That's it from me.
Fantastic. Thank you.
Welcome.
Can I add? So the thing I was going to say after you presented Matt's results earlier, Julie, and it's really relevant given what you just presented there, that this benchmarking exercise is actually really challenging when you're using other people's data, because so much of your effort is trying to figure out what they did, where the data comes from, what it means, and it turns into more of a data curation exercise. So I think the difficulty you encountered there was a large part a data curation exercise, and that's why it was really hard. I think hopefully we're thinking that if the standard should be applied and people should be putting data into the standard, that it's their own data, so they should be familiar with the data, they should know what it means, they should know how exactly a column header that they might come up with would map onto the categories and things like that. So we would hope that a lot of the issues that you encountered wouldn't apply in a real world case. Does anyone else have a comment on that? Carl?
I was just going to say I can show you what I did later, which was my own study. So I can show you some of the issues I had with my own work.
Okay, that would be good as a contrast. Yeah. And I think the comment on the tooling is really valuable too. I think it is always useful to consider how we could help people put their data into the format that we're asking for.
I think one of the ideas brought to me, I really appreciate him mentioned that one. It's like to generate a script to combine a different table into a standard we need. Because if maybe in the real life, like what in one publications, they may have multiple steps, and each of step they're doing their own data. So yeah, if like that one is difficult for like data creators to generate into one big table, actually have a script to assist that step will be much easier for users. That's something we can think about in the future. Yeah. So I like the idea. Thank you.
Great. Any other thoughts on this at this point? If not, I know we had some additional benchmarkers. I know the Open Targets team had done some benchmarking as well. Did anybody want to say a few words from your team?
Yeah, I think I can say a few words. Maybe I can even share my screen for a second. Okay. All right. So there is no presentation here because I didn't have time, but I finished it today in the morning. So my task was to benchmark the peg for the publication describing peg for coronary artery disease. And I got a few issues with the fact, but the most important thing that I come up with here is that the format is, I think, is well defined right now, and I didn't have any issues understanding how it should be, how I should go, how I should start to define. So I first defined the top level feeds in the metadata and describe the variant, gene, etc. And then I was able to just think about the evidence that I got in the publication. So that was quite good, I would say. And I understand everything from there. But there were a few challenges that I had with the data itself, because the first thing that happened to me is that obviously it wasn't my data. So as we talked about it, the challenging of the curation happened. And in this case, the publication was actually doing two different meta-analyses on the GWAS. And that resulted into studies that were then updated in the GWAS catalog. So I had to define one of them, but the issue was that in the knowledge portal, there was a mix of the evidence from both of the studies. So it was kind of hard to go back to one source and to the other one and to understand which variants are coming from which exact study. So the thing that I struggled here is that I think we should well define the unit of the Stroman entity by itself. So from what I understood from the format, it's kind of like it's linked to the GWAS and specific study that we have in the GWAS catalog or some other resource. But I don't think there is much definition about the cohort. Maybe I'm not so sure. Maybe that's wrong, my assumption, because maybe I did not see that in the example file, but maybe I missed it. The other thing that I found very hard in this particular study is that there was a mixture of different evidence categories. So, for instance, there was an evidence from inside of the knowledge portal that defined drag or Mendelian randomization, and the value of that was just a gene that there was no additional evidence. So, going to to get the evidence, I had to get back to the actual supplementary tables and format everything. So I just did a few of them and left the other ones because that was quite long because of 34, I think 34 tables in the supplementary. So quite a big amount. And the last thing I wanted to comment on, I think that we could get some specific fields linked to the specific evidence sources that are required in what we want to see, because I had some struggle to understand what I should actually fill into the evidence. So, for instance, for GWAS, that's fairly understandable for me, but for other evidences that might not be that easy to follow up. I'm again, there might not be this issue if somebody comes with their own data and they describe them themselves. And yeah, I think that's all of the issues that I got. Yeah.
Thank you so much. I really like you point out like the one matrix including the two Java study, because I was thinking that one before, but haven't found a real case yet, and it's a real example for the two Java studies. Could I ask to say is that they understand the Java separately, or just combine the table together when they finish it, or they study
Yeah,
them?
but there was also another issue with those because of the fact that the actual publication comes with the AG19 reference, and then on GWAS catalog site we also obviously have the latest reference from the GRC 38, right? So there was also had to be done. I had to do the liftover for the variants to actually see for instance, the locus boundaries because they were only defined in the supplementary table. So that was a bit of sculpturing, I would say.
Mhm. Yeah, because like in our current assumption is that each of peg matrix should be only is after one JBoss analyzed, so which means it's a one JBoss, one matrix. That is what we're supposed. So actually, I was thinking about is that any possible to say one matrix have a two JBoss? But when I thinking about that one, okay, I was thinking why not they do the meta analysis firstly to combine the two JBoss into one JBoss and then do the downstream things. That is reason I was thinking about that real case, and it's really happy to know actually it happens in the real life. Thank you for that. Another point you point out is really important, and we realized that one before. It's it's colocalization and MR. Both of them have situation to say AI is analyzed. They can combine the different evidence into this one, and which one is the final one to deliver out. That is also something we think about before as well. It's really important. That is really important point as well. Yeah.
Yeah, and thinking of those points again from, a user submitting their own data, you could argue that the two GWASs they should separate and they would provide them as two separate lists, maybe. And similarly with the merging of the evidence, they would have to decide which ones to include. That would be an author choice. But when you're curating it, it's much more difficult to. But they are interesting issues. But I wonder if the two GWAS one is meaningfully two GWAS or in some kind of weird edge case way, or it's just that they happen to co-occur and they could be split. But
So
thank you for your feedback. Yeah.
I think in this context they were So the one was done on top of the other, right? So this was just adding the BBJ biobank. Yeah, it introduced a few more associations in terms of the raw data, but nothing else. But the thing is that most of the evidence afterwards was done on the total, right? So the one with the additional biobank data. So if I were to use the original, so the primary meta-analysis, it would be much less evidence in that case.
Mhm.
Okay.
Great, thank you.
Do you want to be the next, Carol?
Sure, I'd be happy to. Let me share my screen. Okay, can you all see that all right? Great. Is it helpful if I go through the evidence itself very briefly before getting into the metadata, or do you want to just focus on the metadata?
I think we have time if you wanted to go through it briefly. Thank you.
Go very briefly. I just, wanted to point out. So this is a study that I led. We published it earlier this year on Parkinson's disease, and even though I was intimately involved in this project, I think I applaud some of the work of the previous presenters who, it wasn't their own work, and they dug super deep to try to extract all of this information out. I kind of took the opposite approach, which was, if we're trying to have this be something that can easily be extracted, then we should try to see what can be done with minimal effort. So, I could have gone back into my code and pulled out the exact chromosome position ref alt. We didn't publish that in the supplementary tables. And so I think, rather I could have done this, but I thought, okay, well, true to this exercise of trying to figure out what's easy to do based on published literature, this is what was actually easily available from our own supplement. So we put RSIDs in. We didn't actually put in CPRA, which was, interesting. So then we have, gene symbol, ensemble gene ID. For locus, we just had an integer for each locus, 1, 2, 3, 4. I think, I agree. Recommended, it would be nice to have the chromosome, the ranges. This makes sense. We could have done this. We didn't do it. But I think that's very nice to have. We took a very simple approach in this, and it was just based on three things. Approximating, is it the nearest gene? Does it have the top pop score in the region? And are there non-synonymous variants in the credible set affecting this gene? And so I've kind of included both the actual distance as well as this binary yes, no for is it the nearest gene? Yes, no. What's the actual pop score? Is it the top pop score in the locus? Yes, no. And then what is the posterior inclusion probability for non-synonymous variants? And then does that meet the pre-specified threshold that we used in the paper of 50%? Yes, no. And then finally, we have this overall column where we had our prioritized list of genes coming out of that and kind of this ultimate combined result here. I liked the way that this was presented earlier for the GWAS source because this hasn't been deposited on GWAS catalog yet. And I think I could take inspiration from Matthew Paul and put in the medRxiv link instead, which might be better than this description that I've put in. Although I guess we've just heard some reasons to consider going deeper on that if one study has multiple GWASs attached to it. For the genome build, I wasn't sure if we wanted to standardize HG versus GRCH. I found for sample ancestry category, I didn't see something in the guidance that distinguished between category and not category for sample ancestry. So I put this in twice. Maybe that's some feedback for you. I don't know if I've done that correctly. For the ontology, I thought, I'm used to working with YAMLs, and this kind of indented format is fine, but it also might be good to keep things as simple as possible for more lay users. And having this indentation, I think, can be intimidating for people. And, I understand, yes, you could have the kind of plain language description of the ontology term as well as the ontology term itself. Given that you've already got a trait description, I don't know if this plain language field is so useful, and it might just be easier to have an ontology ID attached to that, for instance. I won't say too much more. A few other points, just that I think for what is the nature of your primary variant, I think all of the examples were focused around, is it the variant with the lowest p-value, or is it the variant with the highest pip? Here we ran GCTA Kojo, which does this kind of stepwise procedure of picking variants. And so maybe this is a use case that we haven't seen before that I wanted to bring to your attention, is that the primary variant was selected in this stepwise fashion where you pick the best one condition, pick the best of the remaining condition, which wasn't listed somewhere explicitly, and I wanted to bring that up. I think that's pretty much everything that I wanted to say. I don't think it's so useful to dive into the evidence types. I think that was fairly straightforward for me, but then again, this was a relatively straightforward process where we looked at a very small number of things and binarized them and integrated them without trying to be too fancy.
Can you comment on your integration part though? And how did you represent that?
In the YAML or
Yeah,
in...
did you just just give a text description of what you did, or?
Yeah, let me share that again, sorry. So yeah, proximity, I described all of the columns that I showed you in the Excel spreadsheet earlier, but the bottom one here is the integration. I copied what was already in the template, which was to kind of refer to this as int combined prediction author score. And I filled in a description, and there was kind of some logic of how this was done. Okay, is it the nearest gene and has the top pops? Okay, then we prioritize it. We also prioritize stuff that has a high pip coding variant in it. If there's a clash between those two, we take the coding variant. So explained that kind of in plain text, and then listed the data type as Boolean. I actually wasn't sure if it was Boolean logical, how you want that represented, but in the actual peg matrix, it's listed as a Y or an N.
Right. Yeah. That's good to see.
Great.
For me, firstly, thank you. I really love your reformatted metrics. Really clear to me. So, and also I feel so sorry for the intentions. I know the YAML file is not really friendly for people to create because normally it should be machine generated YAML files. So the reason why we show the YAML file is only to say, oh, this one can be human to readable. It can have all the information you want. But maybe for the people who want to submit the YAML files, maybe it would be more easier with the Excel file or more easier way to do it. So I'm feel sorry for the really painful part. And yeah, and also for the ontology, it's the same thing. Say actually ontology ID is enough for the submitters. But if you are a reader, you are reading the YAML files, the ID can be not really meaningful to you. That's the reason we have the description as well. That is the only reason that here. Yeah.
Yeah. And just to add on you mentioned about the ancestry representation, and it's something we completely neglected to add to the guidance because we've just been working on the GWAS catalog too long, and we assume everyone knows about how ancestry is represented there. But I think we would probably lift that representation, and there's all sorts of documentation about how that is structured and how users can submit that information. So that's completely, we just neglected to put that in the guidance.
And what's the difference between the two categories of
So
sample
basically what you're trying to capture is that you want the user to give you kind of like the most granular thing possible. Like if in how the data was generated, it was described in a certain way, you want to capture that. You always want to capture the most granular kind of piece of information. But then it would be mapped to a more standardized category, which is better for, integration, for example. But
great.
Yeah.
I guess just one thought, Carl, on your to go back to what you had said about human readable versus, ontology-based phenotypes. I'm wondering if we're thinking about doing something like what Abdurrahman suggested eventually, right? Creating an interface for people to do parts of this process and to automate parts of this process, if that might be a place where we could help automate to reduce the burden on the submitters.
Yeah, I mean, I think if you submit an EFO ID, then you can find the plain language description from that, right? So that's kind of why I brought it up. I thought, if you provide the ID, that should be sufficient to fill in the rest of the gaps. And then you still have your trait description column where you can say, well, it's not actually Parkinson's disease. It's, Parkinson's disease in people younger than 50, which is quite unusual, whatever, but you would map it to that ontology. And then you could have it automatically linked to the plain language term for that.
Yes, exactly. I think in the future world, that will be like exactly what we want in the future. And just currently, the standard, we want to say if the information is enough to describe a matrix or not. And the next step could be develop a UI or tools for that, assist that step. Yeah, definitely we will apply that. Yeah.
But I think this also shows that dealing with your own data is a little bit easier than trying to create somebody else's data.
Yeah. Okay, I think because time limitations, like I already share my slides in the folder already. If anyone interested to see the details of my benchmark results, just feel free to go through like the slides on our folder. And because we have like the Aoife to talking about tag list later, and Julie will touch the ASHG again. So I will hand over the time to the Aoife next.
Well, before we move on, maybe if anyone has any more general comments about now that they've heard, everybody else's experience of this exercise, if anyone has any comments about how their overall impressions or in particular like the open targets folks, if how this differs from what you're used to using in your data representation, for example. It seems like we've encountered a couple of high-level things. Curating someone else's data is very challenging. But apart from that, this seemed to work reasonably well for most of the data sets that we've seen. Does anybody want to add anything at this point? Okay. Right, me next, right? We will take all of this and kind of synthesize it, obviously. And thank you everybody for sharing. But before wrapping up, we just wanted to talk a little bit about specifically peg lists, which we haven't really addressed very specifically at all. So I'm going to just touch on that. Thank you, Laura, for making most of these slides. Laura's not feeling well, so I'm going to present them for her. Let me just try and share my screen. And everybody, oh, that looks like the wrong screen. Is that the wrong screen?
Yeah, wrong screen.
Wrong screen. Any better? Yes, that's the correct screen. Great. Okay. So yeah, I just wanted to touch on this because we haven't touched on it in a little while. So I think the matrix is in pretty good shape, but we should just touch on the differences between lists and matrices. And so this is the slide that we presented like way back at the start, talking about what is a predicted effector gene list. So we have our different evidence streams. We have the matrices that we've just been discussing, which show like all the detail of all the different evidence types plus the integration stuff. Then moving on from that to have some kind of distillation of these massive tables, a PEG list that has really just the genes listed that the author is saying are associated with are the effector genes for this trait. I'm not going to get onto gold standard lists, which would probably be you know, a compilation of different PEG lists. So we've got the PEG matrix, which is the structured, you know, the table with all of the evidence types. We've got the metadata, which describes that. The way we're thinking about PEG lists now is like a high-level summary, like a distillation of the best prediction for each GWAS variant in that matrix. And it somehow represents the different evidence types, and it includes a score, a rank, like some kind of integration, which is the author saying, this is the gene I think is relevant for, is the effector gene for this variant. So that's what we think are the key features of a PEG list. And a list it should always be related to a matrix, right? So this is what we're thinking in terms of criteria. That, like I said, it's a high-level summary, like a distillation and a simplification of the results from the matrix. That list should be supported by a data matrix, and therefore the column headers should match the matrix headers. The column headers for the list should match the ones for the matrix. It lists the most strongly predicted gene per variant, per locus. Have this asterisk here because this is a key question. Should it include the highest ranked gene per locus, even if that top gene isn't particularly strong? Or should it only include genes that pass a specified threshold that are like really strongly predicted? Oh, sorry, Julie.
Oh, Aoife, your slides aren't advancing.
They're not advancing. Okay, they're advancing for me. So do I have a PEG list criteria? You can't see that?
I see your title slide.
See my title slide? Okay. I don't know what's going on there. Can you see it now?
Yeah, that looks good.
Okay, let's just go with this. Right. So this was the second slide I showed you. You didn't see this? Nope. Okay. So this was a reminder of the matrices, and then the lists are a kind of simplification of that, basically. So this is what we mean by a PEG list. We have the matrix that we've already talked about. We have the metadata that describes it. And then the list should be derived from the matrix. High-level summary, lists the best prediction for each variant, represents all the evidence types, and includes a score and integration. So I was just looking at these more detailed criteria for the list, the summary, and this is the key question, I think. The list should include the most strongly predicted genes on the list, but should that be the highest ranked gene per locus, even if it's not particularly high, or only genes that pass a given threshold? That's a question. All the evidence streams or different evidence categories that are considered should be represented, and the presented data, well, should be, so this is about the evidence rather than the summary, the output, should be limited to whether evidence again passes a threshold. So you can say yes, no for supporting evidence for EQTL data, for example. And an integration or some kind of conclusion must be provided. So you can have an evidence matrix without any integration information. We're saying that for a list, you should have some kind of integration so that there's some conclusion, and it's not just the underlying data. And that format should be defined in the metadata. We're very short on time, but just to show you some examples of what we mean. Sorry, let me do this. So this first one, I mentioned that the inclusion of thresholding evidence streams. The most simplistic way of doing it, of showing that, would be not even to consider whether evidence streams are significant or not, but just that the author looked at these evidence streams. So you could have a GWAS variant, the predicted effect to gene, and say, yes, the author looked at all these evidence streams and here's their conclusion. Or you could require a kind of threshold for all of the different evidence streams. So that, say, you could see by looking at this that this evidence stream passed a threshold, these two did, but this one didn't. So you have more information about the underlying evidence, but again, the same conclusion in the integration column. You could represent this in other ways, collapsing the different evidence streams within a category. So as you can see here, this one has two different EQTL tissue sources. You could collapse those and just say, yes, there was some data that passed threshold for QTL data. And you could grade that evidence based on how much evidence there is in that category. So we're right up to time, but these are the kind of things we're thinking for lists. There's obviously a couple of questions around thresholding. It's pretty challenging to ask authors to threshold different evidence streams. Should we require thresholding for the actual inclusion of a gene on the list? Or should it just be your highest ranked gene on a list? How to deal with missing data, and whether it can be called a list if there's no underlying matrix. I guess these are all questions to ponder for the next meeting, since we're 2 minutes away from the end, but happy to take any last minute comments on that. We can share these slides and everyone can ponder these questions. So I think, like Julie said, Mackenzie will follow up on scheduling. And it'd be great to hear from you all on that. If anyone has any thoughts they want to share by email, please do. But we will be looking at synthesizing all the benchmarking exercises and all your input from them.
Thank you all so much for taking the time to do the benchmarking as well. This is exactly what we were hoping for.
Yeah.
All right. If not, thank you, and we'll follow up by email.
Thank you, everybody.
Thanks so much.
Bye-bye.
Yeah.
Bye.
Yeah.